here to Dr. Hell. Thank you.
All right, thank you, John. And hi, everyone. My name is Alfred Huo, and I joined the UF in 2017 as a research scientist and as assistant professor and plant breeding. So my major activities is to use molecular biology and biotechnology tool for the research and the plant breeding. Today, I also noticed we do have a very diverse audience here. So I'm trying to briefly discuss the research activity and the breeding activity in my lab, and mainly focus on the following talk here. All right. Something wrong. Oh no. So here's my outlines here. First, I'd like to overview the breeding history and trying to educate everyone. And so the history, the long history about the human being in breeding, plant breeding, and followed by research and breeding activity updating in my lab. And so let's start it with the plant breeding milestones. So actually, human being has been conducting breeding activity more than 10,000 years. So in the beginning, the human being performed selection based on the natural variation. So this selection process also called crop domestication. And a human being, by that time, actually, the human being has no idea about science-based plant breeding. And only until about 200 years ago, and a human being started, the science-based plant breeding, the one we call the crossbreeding or hybridization breeding. In the 1940s, and the human being started the mutation breeding through the physical or chemical mutagenesis. So in the early 2000s and in early 1990s, we started the genetic engineering. I believe the first released GMO crop is BT cotton. So in the early 2000s, and scientists started to develop the molecular assisted selection for the plant breeding, mainly because of the genome sequencing is technology has been advanced, and also the cost is really cheap. So in the 2016, and the people and the scientists from Pioneer Company reported the first genome edited crop in corn. So here is the plant domestication. So plant domestication is a selection process carried out by a human being to adapt plants to their own needs. This selection process solely relies on the natural genetic variation caused by natural power, such as UV light or other environmental stress. There's no crossing, no X-ray mutagenesis, no gamma radiation mutagenesis. So basically, this started with wild species and pick up the best plants with the desirable traits. For example, large grain, higher yield, and less bitterness. So some of you may notice, may guess what plants they are. So actually, our modern current crops were domesticated from these wild species. So I think some of the audience can guess what they are. Here is very easy. This is sunflower, and this one is lettuce. This is tomato. Actually, this is the corn. So you can tell it's really, really different from the ancestor line. So here probably is harder, a little bit harder for you guys. The first one actually is watermelon, and here is banana, and here is a carrot and eggplant. You can tell it's very, very different from their ancestors. And for the hybridization breeding, basically two different parents will cross each other. The new genetic variation will be created. So generally, the hybrid will have characters from both parents. So for example, and if we mate a pug and a beagle dog, and we will create a mixed breed with both pug and beagle traits. And look at the face has mixed features from both dogs and also the body. So basically the hybridization breeding is introduced the desirable trait from one parent to another one. And the early on stage, the selection is fully relying on the morphological characters. So in these days, molecular markers selection for the current hybridization breeding is heavily used to speed up the selection process. So the hybridization breeding is heavily depends on the genetic variation. So what that meaning? So, and without any genetic variation, for example, you can tell from this potato and tomato and wheat, the barley, the trait is morphologically is very, very different. And without this difference, it's really difficult for the hybridization breeding. If we want to create a novel hybrid, we generally pick up the two different contrasting parents. For example, one has very large fruit size here. Another one probably from a wild species with a strong disease resistance. We are going to cross both parents. We expect to have hybrid with larger fruit and disease resistance. However, one common issue for hybridization breeding is that you will also bring in many undesirable traits. When you cross one elite variety with another wild species. And in this case, so we have to using one solution, this is called backcrossing. So when you get this one, the hybrid, you will backcross with one parent. Usually it's the elite, the best variety, for example, with higher yield or good fruit quality. And you backcross several times. And in the end, you will have only a piece of DNA or genome fragment from another wild species parent. So let's take the sweet corn as the example. We have parent A is highly tolerant to the drought, but the quality, the yield, the quality is lower. But another parent is sensitive to drought. The quality is higher. For example, the yield is very high or very sweet. And when you cross both parents for the first generation, you will have half from parent A, half from parent B, which is not very good because you don't want a lot of information, genetic information from parent A, only for the drought tolerance. And in this case, you will backcross with parent B several times. So after 4 generations, eventually you will have more than 95% the genomic information from parent B, but only a piece of fragment from parent A. So what that meaning is it takes a long time. This process is very, very lengthy and also time consuming and laborious. So let's discuss the advantages and the disadvantages of hybridization breeding. The advantages is for the hybridization is that we do not need to know a lot of genetic and genomic information for both parents, and the hybrid products are not regulated by USDA. Basically, is ecologically and environmentally friendly. And the selection is mainly rely on the phenotypic difference. And you can tell here is very large fruit or disease resistance. You can pick up that one for further evaluation. But they do have several disadvantages. And for example, it's heavily rely on genetic variation. But you know, in some plant species, just because of inbreeding, is heavily inbreeding problem. They don't have a lot of genetic variation. The variation is very narrow. And also is time and labor consuming, particularly for the quantity of traits. It's very hard for selection because controlled by the multiple locus, multiple genes. And there are also, they have crossing compatibility. And when you cross one parent with another parent, they cannot produce any seeds because even the pollen can germinate well. So this is partially because the embryo developmental defect. Lastly, is there's one issue is commonly found in the hybridization breeding. They call the linkage drag. So what the meaning of linkage drag is when you introduce desirable traits, you always found some undesirable traits. For example, when you introduce good flavor, but has disease sensitivity, or you increase the disease resistance. But the tomato, for example, the flavor is really, really bad. So next we are going to talk about the mutation breeding. Actually, mutation breeding and is very popular in the 1940s, and the spontaneous mutation is caused is continuous source for the natural genetic variation, and mainly caused by the environment stress or other environmental conditions. For example, the UV light from sunlight, or the insertion of endogenous mobile DNA element transposing. I'm going to discuss a little bit more in the later slides. And in 1940s, the people using artificial mutagenesis, mainly using chemical or physical mutagens to induce the mutations. For example, the gamma irradiation and X-ray, etc. And the artificial mutagenesis could be very efficient compared with the spontaneous mutations in the nature. But many non-desirable mutations will be created, will be generated. So for the traditional mutagenesis using the chemical or physical reagents, and the mutagenesis is really hard, it's randomly. So the mutation is random, like we are using the shotgun to shoot the plate here. Randomly is really hard. So the random shooting can happen everywhere, you can tell. Actually, your real target in the both eyes here in the blue plate, and you can consider the blue each plate as one chromosome. I believe some of you will understand the chromosome in the plants. So what you can do, and you can use in the backcrossing to clean up the genome, and as I mentioned in a couple of slides before. And so the meaning is you take maybe 4 or 5 generations to remove majority of non-desirable and bad mutations from this hybrid, the mutant. And in most cases, mutagenesis and dangerous can cause weird and undesirable phenotype. For example, this is caused by the water pollutions and it's really bad mutations. But the scientists using the physical or chemical mutagenesis and to create some useful varieties in plants during the plant breeding. Here is some examples. Here's seedless orange and salt tolerant rice, and these different colors flowers and also the foliage with different leaf patterns. Of course, they do have some advantages and disadvantages. And as a traditional hybridization breeding, we don't need to know the genetic and genomic background of the plant materials. And induction of a desired mutant, that is, you can introduce any fate that cannot find in the nature. The product is not regulated by USDA, and basically ecologically and environmentally friendly. The procedure to make the mutant library are relatively simple and fast. So basically, you give them plant materials, you treat with gamma irradiation or X-ray or some chemical reagents. But the mutation generally is random and unpredictable. There are always good mutations come with bad mutations. So in order to find the good one that you want, you need to create a large mutant population, or we call the mutant library or pool. And it's very costly and very slow for the selection to clean up the genotype, the genome. So next, I'm going to move to the genetic engineering. Probably all know the GMO crops. So since early 1990s, we have started the genetic engineering of many different crops. So the first released crop, I believe, is insect resistant cotton. Since then, we have huge controversial issues on the genetic engineering. So companies like Monsanto said they are safe, but the public do not accept the GMO foods even today. In some countries, they do accept the ornamental plants, for example, petunia or roots. And personally, I have no comments on the GMO. Today, just briefly update the current status of GMO crops in the world. So here, at least 30 different crops. Just let's take a guess. How many of them have been genetically modified and proved by different governments and grown on the earth? So the truth is all of them. So all of them have been modified. You can tell it's including apple, pepper, and papaya. But how many of them in the United States have been approved? And we have, it surprised me too, and we have 203 approved varieties. I believe this number increased a lot in past 5 years. This data is generated in, I think, in 2016 for 21 different crop species. We have a lot of different GMO growing in the United States. So what's the major traits modified through the GMOs, through the genetic modification? The major traits is a herbicide tolerance. And we have 12 herbicide resistant crop species on the market. And the second followed by insect resistant. We have a cotton, corn, potato, etc. Totally 7 crop species for the insect resistance on the market right now. So here are some examples for the GMO product. And look at here, the left is a sustainable plants for the rain spot virus. They destroyed all the papaya plants. The right part is rain spot virus resistant papaya, golden rice, and the BT corn, and omega-3 enriched soybean, and apple anti-browning, and apple and potato. Here, Arctic. So you probably heard that one on the market several years ago. And the blue rose developed by Australia, and also released on the market in several countries, including the United States. So here are some top countries that are growing GMO crops. Actually, USA has been ranked the top one, and about 39% of total world planting area, followed by the Brazil, Argentina. And probably you know that the major GMO crops in the United States are corn and soybean, mainly for the feedstock and the ethanol. And the total, actually in past 10 years, the total planting area has increased almost 10 times from 1996 to 2016, mainly in the developing country. You can tell this red line is developing country, the blue one is developed country like US. Okay, so the advantage is for the genetic modification or GMO. So when you transform a gene or limited number of genes, it can precisely modify specific traits of crop, not like a traditional mutagenesis or hybridization. You can introduce undesirable. This is very specific. They can create desirable traits that are not found in the natural plant population. So, but the major problem is you have to know the gene function, or you have to know the genome information of this gene. It's very costly to generate GMO crops because the procedure is very complicated and requires specific technical skills. Also heavily regulated by USDA and take years for commercializing and very hard for consumer accepting. So the latest innovative technology we call CRISPR genome editing, and CRISPR technology was initially discovered in the bacteria in 1987 by a Japanese group. So basically, bacteria using CRISPR to defend the attack by virus. So only recently, scientists from UC Berkeley and Harvard University have modified CRISPR for different research studies, including plant breeding. And there are some major features for the CRISPR genome editing. So here is some of them, and high precision, high efficiency, broad application. And although the procedures for CRISPR gene editing product is identical to the genetic modification, so which means it's identical to the GMO procedure, but the final products is similar to the traditional breeding, so which means that they are not GMO, they are GMO-free, DNA-free product. So if we compare the traditional mutagenesis and with gene editing mutagenesis, you can tell the mutation created by the traditional mutagenesis random, as I mentioned before, so you can see everywhere. But for the mutation created by the genome editing, it's very direct to the both eye, it's very specific. And we don't need a lot of effort to clean up the plant genome. So why the CRISPR genome editing is can create precise mutations? So because this CRISPR technology using a complex toolkit. Here is a toolkit, and they have two parts. One is here is target, we call it a guided DNA sequence. So you can assume this is a photo ID. It's like a driver license. Here is, we call the cart, is scissors, molecular scissors. So basically, and the toolkit can use this photo ID, can recognize the unique location. And they can search and target the specific target gene, and the scissors, molecular scissors, can be used to create a NIT to turn off or turn on the gene function. So regarding the efficiency, and here is one example. comparison of efficiency between the traditional breeding and the precision breeding through the genome editing. So for the traditional hybridization, you have to go through 7 generations at least. So for the selection and the evaluation, each generation, you have to pick up the right one and evaluate the phenotype again. And for each generation, you need to grow at least 1,000 plants. You can imagine how many plants you have to grow. It's a lot of money, a lot of effort, and timing, and time you have to contribute. On the other hand, for the genome editing, so you need only 2 generations. Okay, each generation, you need only 50 plants. That's significantly different. And the genome editing can significantly reduce the number of crossing cycle and significantly reduce the plant number of each generation. Generally speaking, so if you're using genome editing, you probably can release your first cultivar in 4 years. But for the traditional breeding, will take around 7 to 8, 9 years to release the first cultivar for the field crop, okay? And for other crop, maybe 4 years or 5 years for the vegetatively propagated plants. For the GMO, will take around 13 to 20 years. Why is it takes so long time? Because mainly for the deregulation of GMO crops. The USDA and the FDA require a lot of trial tests for the GMO. Regarding the broad applications, so one gene called MLO1, it controls powder mildew sensitivity and has been identified in many crop species, such as wheat, tomato, petunia, and lettuce here. And some scientists has turned off the MLO1 gene in tomato. You look at here. So as a result, and the tomato, the gene-edited tomato plants become strong resistant, display strong resistant to the powder mildew. So on the other word, so we can using the genome editing to turn off the MLO1 gene in many different plant species. And this technology can be used in many purposes. So although the procedures for the CRISPR gene editing is same as the genetic modification or genetic engineering, but the gene edited product is non-GMO. Because the mechanism between the two technologies are very different. For genetic engineering, you have to put a foreign DNA or transgene into the plant genome. So the transgene will generate the trait. For example, the BT gene for the insect resistance. Without this transgene, you cannot observe any insect resistance. But for the CRISPR gene editing, the transgene here is the Cas9 gene, only acts as a doctor's tool. You think about the doctor, physicians, the Caesar, and the doctor using the tool to cut the damaged tissue out. And then the tools will be discarded. You can look at here. This is created and make the mutation here. And here you have transgene will be discarded through the segregation. In our lab, we have developed some tools. And in this tool, we have put a fluorescent gene in there. So we're using this tool to edit the lettuce. Usually the lettuce seeds cannot germinate in Florida because the temperature is really high. The high temperature will inhibit the seed germination. Look at the gene-edited lettuce. They are able to germinate in the high temperature, but we don't know which one is non-GMO lettuce. So we put these germinated seedlings under the UV light. We are able to see, okay, this one has fluorescence. We are going to discard because they have a transgene, they have a foreign gene in there. But for this one, they are non-GMO product that we are going to use. So it's the One important question is, this genome edited crops regulated by USDA? Actually, in March 2018, USDA has released a statement said genome edited plants will not be regulated as long as they are not for the plant pest and also the weeds. So, but still there are some challenges and limitations to use CRISPR gene editing in plant breeding. So first, we need to know the genome sequence information. And second, we have to need an efficient plant transformation pipeline because we have to put the CRISPR complex, the tool into the plant genome in order to get the mutation. So, and also we still don't know if the public accept the gene edited product, if there's any ecological concern. So next, I'm going to briefly update the research and breeding activity in my lab. When you buy a lettuce and store them in your fridge, and several days later, you probably observed this. Okay, the lettuce lose their color, and also they're getting browning and get rotting. And in our lab, we're using gene editing to create a mutant. And then, this is a mutant, and you look at it, the leaves is dark color compared to the wild type. So we take the leaves and we detach the leaves, put these leaves in a dark condition and at room temperature. So the purpose, we try to develop some mutant letters can, have prolonged shelf life. And we test if they have a prolonged shelf life under the dark condition. So you can look at after 7 days, and the wild type turned yellow, but the mutant stay very green. But after 14 days treatment, and the wild type leaves looks like very, very ugly, and the mutant leaves still fine. And we also using gene editing to increase the organ size. We did in the lettuce and also in the snapdragon and petunia ornamental plants. Look at the leaves, the mutant at the different growth stage. The leaves size is really large compared with the commercial varieties. And you can literally to see it's almost 3 times larger than the wild type commercial variety. So we also added a gene called a variegated variegation 2. And after we added this gene and we observed the variegated pattern in the lettuce leaves, the purpose is we trying to translate this method to develop some foliage plants in the future. And next, I'm going to talk about the plant breeding with transporting mutagenesis method. What's transporting? Transporting, also called transposable element. Basically, it's a sequence of DNA that can move from one place to another one in the genome. Look at here, the green one is transporting, and our body has a lot of transporting, so probably you don't know about that. And especially in the corn, they have a lot, 90% or 70% of DNA, the genome are transporting. They can jump, so they are not stable. They can jump from one location to another location and randomly jump, okay? After they jump, they can insert into the different genome to create a mutant. So using this method, we can create a lot of different mutant. So actually, there's many plant mutant around you that you may not notice, and it are created by transposing mutagenesis. For example, this orchid with the red purple pattern is caused by transporting insertion, and the purple cauliflower and the blood orange, navel orange, and also the white carbonate grapes, a weird sunflower head here, and here the yellow tomato and yellow color tomato, and also Rama oval shaped tomato that you always buy from market, all caused by transposing induced genetic mutation. So probably you all don't know about this. And actually, in our lab, we have received some Snapdragon germplasm and from John Innes Center in the United Kingdom. And look at this one. This is a wild type, and we call the GI7 line. The flower is red. But for this one, we call the GI2 line, is white. What happened with this flower? Because they have one transposon and insert one gene to block the anthocyanin, to block the pigment production. As a result, the flower become white color. Very interesting. When we grow this plant, this plant at 20 Celsius, or above 70 Fahrenheit degree, the color will be white. But when we grow the plant in the 15 Celsius or around 60 Fahrenheit degree, and the color, become red. What happened in there? So because the transposing can tell the difference, the temperature, when the temperature become cold, they are activated. They jumped randomly and they insert somewhere else. As a result, the anthocyanin will be restored. The pigment will be reproduced again. The color, the flower color will become red. So what that meaning? So when we saw the color become red, we know that mutation happened somewhere in this plant. So from this flower, we are going to harvest the seeds and grow them, and to look into the some phenotype. So, for example, using this method, we create some mutant. Look at this one, is dwarf and head cluster mutant. So it's very different from the commercial variety. If this is the top view, this is side view. I think this is very pretty. And this one is trumpet flower mutant. You can tell it's very different. This is red color, and also this is a white color. This is the same. They also change the color. Here is heat and drought tolerant mutants. So basically, we put the commercial variety and our mutant in a greenhouse. We just don't give the water, and for 15 days in the Florida summer in July. And you can imagine how hot the greenhouse. We reached around 100 degrees. And after we rewater the plant, only the mutant can survive. And here is some amazing plant that my grad student took the picture. Look at this red leaf color, the mutant here. And this is a green, this is red leaf mutant. And besides the Snapdragon breeding, we also do some heat tolerant begonia breeding. And in past 3 years, we collect more than 70 different begonia germplasm from everywhere, botanical garden and USDA nursery. And some of them is amazing. Look at here, the red color, red leaves begonia, vexed begonia. They are highly heat tolerant one. Some of them from Hawaii Island. But on the other hand, you look at the commercial variety, they cannot survive in the summer, Florida summer, because the storm and high humidity and also the high temperature will kill them. So currently, and my PhD student and biological scientist have conducted a lot of pollination, and we have evaluated more than 5,000 hybrid seedlings. Look at here in the July, they are survived the Florida summer very well. The most commercial variety cannot survive Florida summer. And here is heat tolerant hybrids is a potential variety that can be released in the next 2 years. So here is my big team. I have around 9 people in my lab, actually more than 9 right now. And here is 3 PhD students. Chi is, and also the postdoc and working on lettuce. And the PhD student, Frank, working on Snapdragon. Julian is a PhD student working on Begonia. Toriko, and also the Manish is a postdoc working on tomato. And my lab manager, Matthew, working on the Begonia, pollination, crossing, and selection. And the postdoc Grace working on the tobacco and develop some whiteflies resistant trap plants that I have not talked about here. So I think I'm going to end up with my talk. Would be happy to take any questions from you. Any question? I cannot hear.
We do have some in the chat room, Dr. Hu.
Okay, look at, can you speak to the number of genes modified by each of these techniques? Hybridization, mutagenesis. We have this odd term, GMO, in fact, that happens to be among the modification techniques that changes the fused gene, right? Okay, all right. For the we just for the traditional breeding, and basically it's hybridization breeding and also the traditional mutagenesis, and using the chemical or physical reagent, they can change a lot of genes. You are right about this. And for one times, you can select different genes, different traits. For example, you can pick up the good yield and also the plant architecture or dwarf or plants together simultaneously. For the in recent years, for the genetic modification and GMO technology, as well as genome editing, we also can manage multiple genes, not only one genes. So, for example, for the CRISPR-Cas9 genome editing, some labs, some research groups have demonstrated can modify more than 10 genes at the same time. So which means you can look at the 10 different traits. Theoretically, you can look at the different traits. And there, for the good things for the precision breeding using the CRISPR-Cas9 genome editing is very precise. And you can target specifically on some traits without disrupt other traits. And in the traditional breeding, like hybridization breeding or mutagenesis, is extremely difficult to achieve this goal through only one step or more, several steps. But the gene editing can do that within one step or 2 steps. So it's much easier. The only thing is for the genome editing, it still is very expensive, very costly, because the procedures is very difficult, very technical demanding. Impact pollinators. I think the GMO, to be honest with you, I don't support the GMO But for the genome editing product, they don't, in my opinion, they don't have any negative impact on the pollinators. So, what's that happening? Because, when you work on using the genome editing to create the new product, and they don't introduce any foreign gene in there, and they just make their own, create a mutation. The mutation created by genome editing and is almost same as created by traditional method like chemical or physical mutagenesis. The only difference is the gene editing is more specific, and they don't introduce any foreign gene in there. So like BT or herbicide resistance, they're using their own DNA inside the plant genome. But for the GMO, yeah, it probably will affect the pollinator. That's why there's a huge controversial issue on the GMO. The people said many more studies showing that GMO has negative impact on the ecological part. So particularly for the pollinators. So how much the work in your lab seeks to address conditions unique to the Florida, such as heat, so humidity. The major research interest in my lab is deal with heat and stress tolerance. Basically heat, salt stress, heat stress, and also drought stress. And why I said the drought stress, and because when we develop some varieties and for the landscaping, we don't want to water very frequently, and also the growers, even the home gardener, don't want a lot of water or irrigation for their the backyard plants. So that's why we also interested in the drought. So the major research in my lab is almost all about the heat tolerant. Of course, we also work on some other trees, such as the post-harvest and the shelf life, to improve basically to improve the plant quality through the biotechnology tools, mainly through the genome editing. So for lettuce, is there any drop off in the nutrition in the storage? I have no idea about that, but this is right now we have generated a mutant. That's a very good question, and we definitely will evaluate if the lettuce will be more tolerant to the bacteria infection. So we do have a lot of E. coli bacteria infection, in sometimes, and you always heard news, hear the news, and we got the bag, especially for bagged the lettuce, they got infected by bacteria, and we believe our mutant has strong resistance to the bacteria. And why? Because they have the detached leaves has a longer shelf life, just because they have less degradation in the chloroplast. The chloroplast, is give you the green color in there, and will maintain the leaves much healthier compared with the other commercial variety. Do lettuce then hold better in storage, also keep their nutrition longer? And we are going to test the two conditions. One is in dark, because our mutant is very sensitive to the darkness. When we store our lettuce in the fridge, basically it's a dark condition. And on the other hand, and we also using the lower temperature for storage. And we are going to evaluate our mutant. It also has improved the quality after storage in the fridge at a lower temperature. So basically, we will do evaluate the nutrition level through some other techniques. So I think this is very, very good questions. So then we are planning to do this. Any more questions? We have very good audience here with very good knowledge, and it surprised me. So I know this, we have a very diverse audience. I'm trying to try my best to explain the new technology recent years are heavily used by different research groups, including mine. But I just have a comment. The traditional breeding is still the major tool that being used for the all the plant breeding. But you know, for the genome editing, it's kind of a complementary tool to speed up the breeding process. For example, I just briefly give a talk here, and the genetic lineage, that's why genome editing is very important. And when you try to breed the tomato, you're trying to breed a disease resistance here controlled by a yellow box. But there's strong linkage, poor flavor, and poor post-harvest quality. And you just get disease resistance, but never get good flavor and better post-harvest quality together with disease resistance. Why? Because they are closely linked to each other. They always be together. So you want to get a disease-resistant tomato, but you have a poor flavor too. So in this case, what you can do, we can use in genome editing and to knock out the poor flavor and to turn off the poor post-harvest quality gene, only maintain the disease resistance in the plant. That's the only thing. Okay, so yeah, I think that's why it's, I just say, okay, genome editing is not a major tool, it's a complementary tool. Okay, so any more questions?
Hi, Dr. Hu. I'm Jody Bro. As you know, we had met before this meeting, and whenever you feel comfortable and everybody else feels comfortable, then you know, you're welcome to do whatever you need to do now.
Okay. Oh yeah. Okay. All right. So you guys can find my website and Alfred Lab, and if you're using Google, and you can always to find my contact information. Any question, and can reach to me through the email or phone, and I would be happy to communicate with everyone. Thank you.
Thank you very much. It was very, very good. We truly appreciate all your hard work and what you gave to us today. Something to think about.
Okay, okay, I just stop here. I'm going to leave.
I believe so. I'm not really sure.
Okay, I can leave here. Okay, thank you.
Thank you. Thank you.
You guys have a good day.
You too. Thank you. Hi, Master Gardeners. We're not, John and I mentioned we're not going to be having a meeting today, except for those of you who are on the Legacy Award Committee. John has, we're going to be meeting right after. So those of you who are in the Legacy Award Committee, the members for 2021 and 2022, please stay on, plus Janie Sailors, and we'll go ahead and have our meeting. Everyone else, John said they'll have a lot to be discussed in November. So we hope to see all of you, and whenever you feel comfortable, we'll see you next month. You can go on. Thank you. Legacy Award people. Let me see. I think this may be. Hello.
Hello.
Okay, I am in a, I'll call you back later. Right now, I'm in a meeting. Bye-bye.
Hello, Jeanette.
Hi, is everybody here?
Hi, Ed. While we're waiting for people, Ed,
Yes.
I don't have that card, and it really depends on using that. So as soon as I get it, I'll give you a call.
Well, we'll have a discussion once the meeting gets started, okay?
Yeah, we still have a couple of people still on here, so I'm not sure whether they're at their screen or not. But if you're not a member of the Legacy Award Committee, we'll see you next month. We should be having, let's see, Jeanette should be here. I'm here. Ed's here. Barbara, Mick, Alicia is going to be by phone, I think. Alicia, are you there? And Loretta and Heather. And I'm just waiting for, I'm not sure how to get people to, take people off of them. We have two people.
This is Loretta. I don't think I'm muted. I'm unmuted.
Okay, you're unmuted. Is everybody unmuted?
No, Barbara Wilson's not unmuted. Barbara, are you unmuted? Nope.
Right, and I have, I'm not sure how to, remove people.
Can you hear me?
Remove, okay.
Yes. Hear you now.
Okay, let me see. I think the only one we have here is Tim. Tim, if you're here, I'm removing you. Bye-bye.
Hi. No, he's just here because I need his computer.
Okay. And Mark, I'm going to have to remove you also, okay?
I'll just stand by for the meeting.
Okay, the only person not here is Alicia. And does anybody have her telephone number? Because she said she was going to stay on.
Whose number do you want?
Alicia, if you could, you're having an echo, Barbara, too, with you.
I'm looking it up.
I think everybody else is here. John will not be here. As you know, he had to leave, and I'm the host. So it was very nice of him to do that. As we're doing this, let me go ahead and get on the screen. And
I'm still looking up Alicia's number.
Okay, thank you. She has problems, and we were talked or emailed today, so she's been having problems with her computer. I know none of us can relate to that. That's all I've been having, and I was been told by someone who came to work. My husband has a new computer. He told me my computer needs to be changed if I want to do the stuff I'm doing. So,
well,
I think he was trying to sell me a computer.
Yeah.
Okay. And then we'll just see if Barb is doing.
Hey, I'm just looking for Alicia's phone number.
Barbara, you've got a terrible, terrible echo.
Hang on.
I think she has two computers going on.
Something's happening.
Yeah, that's what happens when Tommy and I use two computers. Like for this, we're usually together on one because it sounds like a science fiction movie.
Is my echo better?
Yeah.
Yes. Okay. I'm looking up Alicia's number.
Okay, thank you.
Yeah, her phone. You want me to call her?
Yes, please, because she said she was going to stay on, but I only saw a telephone number on here when I was looking.
Her number is, I'm calling her right now.
Yeah. And I told her that we were going to stay on. So just, I told her that. So, we should all be, she should be the seventh person. And let's see, Loretta, you can unmute yourself.
I think she hung up on me. Maybe she's dialing in.
Okay.
Well, we have a quorum.
And Heather, you can unmute yourself too. Unmute. Oops.
Hey there, Heather.
Yay! Yay! And we still have Sam on here, Barbara. That may be why
I have Sam on there because I have some. If I need to go back to the original files, I have them on his laptop.
Okay, great. Okay,
just ignore him.
Oh, okay. Oh, how can we ignore Sam? He answers the phone so nicely.
So We have Heather, we have Loretta. We were just missing Alicia. I'll keep trying to call her. Okay, the only one we don't have is Alicia.
Yeah, and she was very, you know, insistent that she was coming just not too long ago. I mean, you know, this morning. So how's everybody else doing?
Fine, doing good.
Good, I'm trying to get my
Fine.
picture up, but not that you necessarily
Busy.
see me. Who said busy?
Okay, so you,
Got to be Heather.
Heather, yeah. Well, none of us are busy, Heather, just you.
so Alicia is not answering her phone. She probably doesn't know my phone, so.
Heather, we can only see half your face. There it is. That's better.
There it is. I'm leaning over a bunch of papers, sorry.
No, I'm talking about Heather.
Okay.
You're driving your car, Heather?
Yes.
Uh-huh.
I'm almost finished driving my car.
You know, this is a confidential meeting. The person at your right is better not be able to understand your laughing.
Yeah.
Only speak Spanish.
Oh, perfecto mundo.
Okay, so do you want to call this meeting to order?
Yeah, I will.
Jody?
Okay, I'm going to go ahead and call it. Can everybody see everyone right now? And then I'll share, oh, it's still recording. I got to take this off. Yes, we don't need us recorded.